then we will start. Alright, for those who just joined us, if you can help to mute and to stop your video, that will be great. We will focus on Dr. Jaya only today. Okay. How many people here? 25. Well done, everyone. Welcome, welcome. And we will start in 2 minutes. Yeah, just now there's a little bit of technical issue. And I just finished work, honestly. Okay. Let me run the talk poster again. Then, Doctor, we will be starting, yeah?
Okay.
All right. So I will repeat. So I will introduce, of course, greet everyone, and then I will introduce who are you, and then I will do some short housekeeping announcement. Then I say, let's welcome Doctor.
Sure.
Okay, 1 more minute to go. Okay. Good evening, everyone. Welcome back to our first talk of the month here with Sabah Society. And today we have Dr. Jaya with us, who is a senior lecturer, researcher in the field of mycology and plant pathology at University Malaysia Sabah. His research interests are to document, describe, and to assess the species diversity of mushroom in Borneo and to utilize them for sustainable use. Besides, he also conducts several environmental education seminars and workshops for kids and adults to promote the awareness on mushrooms' importance to trees, human, and nature. Dr. Jaya also a recipient of prestigious awards such as the International Association Plant Taxonomy, Charles Kramer Award, and Whitley Conservation Research Award in his field. Now, before we start our talk for tonight, there's a short housekeeping announcement. For those who join us on Zoom, I would like to request you to mute yourself and stop your video, or our technician Rachel will help you to do it. I hope we can. And of course, if you see right now on your, for those who are watching us from Facebook, you will see this poster, donate here, support us to Sabah Society. So we really appreciate your donation. In previous talks that we have received like sum of amount in the society. So maybe later I'll announce the total so that everyone would know the total donation that we have received. So for those on Facebook that not sure how you can donate to Sabah Society, you can see this poster on the right side. There's a QR code. You can use Do It Now, or you can use Boost. Just scan the code just like you scan My Sejahtera, and you can put any amount that you like. Now, back to the talk for those who watch us on Facebook. And the other announcement is the Q&A. So Q&A will be held after Dr. Jaya finish his talk. All right, so that's it. Let's welcome Dr. Jaya.
Okay, let me share my slides. Okay, can view my slides?
Perfect.
Is it okay? Can you all hear me clearly?
Yes, can see.
Okay.
Thank you.
So let's talk about mushrooms stories that who initiated by Professor Corner, who a prominent mycologist, a botanist, I would say. Then he went into the mycology because his work is tremendous for our knowledge on the early taxonomic descriptive species on mushrooms. Because not only plants, but he has also had a huge contribution in the mycology field, especially in the Borneo region. Okay, so the topic for today is the larger fungi of Borneo, which is the Pagler's book with the concept of larger fungi. I mean, it's the Basidiomycete groups, which are the fungus or mushrooms that you are able to see with the naked eye. So why do I put here as EJH Corner's journey? Because definitely he was the the mycologist that we are referring to, his works and his collections, and also all of his descriptive works on species level identification. So as you can see from here, I was very glad that Sabah Society has put the Kukena, the cup fungi photo, as a wallpaper here, which I will talk about it as well later on. Okay, where this is one of the unique and also very common tropical species, and they are pantropical. They can be found in any tropical and subtropical region. Kukena tricoloma, yeah, scientific name. Okay, so without further delay, okay, I think I would like to introduce myself. My name is Jaya, so you can just call me Jaya. And I've been working on fungus since 2004. Yes, I'm from undergraduate, master's, and PhD. So it's all about mushrooms and fungi, especially in plant pathology, and whether it's Basidiomycetes or if it's Ascomycetes. So the kingdom fungi, when I look into the ground or terrestrial fungi, they are beautiful, colorful, different shapes, phenotypes, and they are hairy, non-hairy. As you can see, the margins are different margins. The gills are crowded. Sometimes the gills are very distant, and also the shapes and also the appearances on a different substrates, whether it's from soil, whether it's from log, or whether it's dead wood, or branches, or even on a mosses. Okay, so there are diverse groups of mushrooms. Okay. Is that okay? Can you all hear me? Can you all hear me? Okay.
I think someone just joined, so they haven't muted themselves. Yeah.
Okay.
So please continue. Thank you.
Okay. So the diverse groups of mushrooms harvest diverse habitats, which we call the host or substrates. So they can grow everywhere, anywhere, depending on their conditions and also their favorite conditions like carbon, highly rich carbon, nitrogen. So whenever there's carbon and nitrogen available, yes, you can see blooming everywhere on the ground. Okay, not only just ground, on a rock sometimes, and because the rock types are different groups of fungi, and even water, leaf litters, and also a lot of tree, whether it's living or dead trees. So you can see everywhere different types of mushrooms on a different substrates. Okay, that's the basic to understand how diverse they are in our ecosystem. So for a layman concept of how this fungi forms their own family. So as you can see from the cartoon here, it's a simple educational tool that we have created for the layman type of people that including the citizen science who always the hikers, they will be questioning whether it's a fungi, is a plant or animal. So we come up with this kind of educational tools where it's a plant or animal. So we did not know at the time before the molecular revolution. But now with all the DNA evidence, okay, we are now very sure that, plant and animal is a different kingdom and fungi stands on its own kingdom. So finally, we know that the fungi kingdom is always, that close to the, animal kingdom because they are heteropods, right? So they are heterotrophs. They don't obtain any kind of, they cannot produce own food like how a plant doing through the photosynthesis. So people have segregated the fungi group into a different kingdom. So it's amazing to see how when I came back from US, which is in 2015, people still don't believe that fungi has its own kingdom. They still consider under plant. Even lichen is a different thing, which is associated with different entities like algae, cyanobacteria, and fungi. It's a mutual combination, symbiotic relationship between these three entities, then they form the fungi. But fungi is a totally, whether it's a Basidiomycetes, Ascomycetes, or Glomeromycetes, all the chytrid fungi, early diverging plates, everything is under different phyla within the fungi kingdom. And there are 5 phyla within the kingdom of fungi, and you all have, you all have seen these in a documentary kind of in Netflix, right? Okay, so that would educate you all to understand better the basic of fungi. So, for me to show what is the nutrition mode of fungi, we have to understand the basic concept of nutrition mode. Because when you are trying to understand whether they are mutual, whether they are parasitic, whether they are saprophytic, or they are generalists, we do not know. But with the collections of many different temperate forest fungi, tropical fungi, now people have categorized this based on DNA evidence. Okay, the plant-associated fungi usually they forms a mutualistic or symbiotic relationship, especially with dipterocarps or even eucalyptus. Because in different countries you have different trees. Okay, like for example in the US you have oak trees, pine trees that associated with only several types of fungus. However, in tropical, it's huge, diverse within the Dipterocarpaceae as well. But other trees, as angiosperms or gymnosperms, they still have different types of fungus. But we do not know exactly how the cataloging of host trees and fungi, there is no documentation. So in our studies, we are trying to collecting all this information, what type of tree and what is the fungus. So at least we could create a database for this to educate other people. Or even when you want to plant a tree nursery and let's say the tree is not growing. So with the help of mycorrhizal stuff, we could boost the tree growth as well. And basically when the publication on the nutritional mode of fungi was established for Basidiomycetes, Professor David Hibbett and the group of Binder, they have identified how they can switch according to the the evolutionary scale. Okay, because from the generalist they can become a mutualist, or they can become a parasitic, or they can become a saprophytic, or they can be just a generalist group. Okay, so there are 4 different modes over here. But you must imagine all these mushrooms, the Basidiomycetes or Ascomycetes or even the other phyla, okay, they have this, the loss and the gain throughout the evolutionary scale about their parasitism or saprophytism or mutualism or even a generalism. So these are the things that shapes the fungus to become more, more important for our ecosystem. So this is the main point that you all have to get it when you talk about nutrition more. Okay, so there are many different forms of fungi available out there, which are the zombie fungi, which is mostly parasitic, but still the zombie fungi, not all the zombie fungi is parasitic, because some evidence shows that they were generalists and then they become the parasitic, and then some they are saprophytic as well. On beetle, you can call this name saproxylic beetle. They just grow there. Okay, and Labolobiosis is also another parasitic form, apart from the zombie fungi, which only host dependent or whether they can be found on anywhere. Okay, and symbiotic, when you talk about the most important example that we can see is from Borneo is the termite association, because here in Malaysia, the recent one that I described one new species was from my UMS campus. Okay, and the Termitomyces gilbus was the host. So we need to know what is the species of termite and what they do the cultivar on. I mean the cultivar species of the fungus we need to identify. So when we did the molecular DNA and the morphology, we found out that it's unique group compared to the known Termitomyces. And the same thing goes for saprophytic. Saprophytic is a huge number of fungi which still needs a lot of studies. But of course, until I retire, I cannot finish this. But my students are and collaborators, they are all looking into different angles. So we are doing this in a collaborative way to address at least how many types of mushrooms available in the region of Borneo. So these are the mycologists in Malaysia, where some of them are actively engaging on the research on mushrooms, and they have previously past researchers and also some current researchers. Sometimes, as I say, taxonomy wise, there are only 2 people available here: Tutor, me, myself, and Dr. Ismail. So otherwise, the application of fungi, yes, people have done on many different angles on biotechnological application, medicinal, pharmaceutical. But taxonomy-wise, the systematic revision is very less, and we do not know how many species are there for Malaysia exactly. So this is another highlight of the paper that I found out, that global diversity and geography of soil fungi. Why soil fungi? Because the terrestrial fungi usually on the ground. So when they analyze this metagenomic analysis, so as you can see from here, the global wise, how many percentages and how much of huge gap available for tropical moist forests and tropical montane forests. So this is something that we need to address as a mycologist. Yes, these are the concerns that we have to look into it, and then we study the organisms and also their ecology and also their importance. Okay, so in Malaysia, mostly we have to know if the species name and also what is the benefit of that species. How do they become an important in the ecology? So that way we can sell the importance of mushrooms to many people. Okay, as a history background, okay, when I started my PhD, so the prior studies was actually from Panus, Lentinus. I will show all these pictures later on for you. But here, when Horner and Pagler, okay, they started with 2 different classification on few groups of mushrooms. But here I'm talking about the Lentinus, Panus, and oyster group, which is the Pleurotoid. Okay, Pagler has classified them all under one subgenus. Okay, but however, Horner has grouped them into 3 different genera. This was molecularly proved that Horner was right, actually, because Horner has studied their morphology, has studied their phenology, everything in complete, and he has concluded that all 3 different groups, which are the Lentinus, Panus, and also the oyster groups, are totally 3 different genera. So from there, we have addressed and studied on Lentinus. So I did my PhD on the Lentinus group and also the oyster group, which are the mostly morphological characters are very confusing, and also a lot of demarcation on the classification. So in 1981, when he started, so it evolved to become a lot of studies and how many species that have been described all of this while, and still the studies are going on. So this is the journey of where Horner started, in a nice way of how to classify these mushrooms in a correct genera. Okay, but his identification is very reliable. I mean, it's pretty good actually. I mean, I see Serena is here. So, I mean, from Singapore side, we have worked together as well. So she knows that how to appreciate faunus classification as well, compared to pegless classification for just within these three genera. Okay, but what about other genera? So we will go on to other genera as well. So what we do actually in our lab. So basically we do a lot of field work, sample collections, and we do a lot of photographs, GPS, notes, and all the micro characteristics. Sometimes I do receive samples from the citizen science where when they are going for hiking, so they send me the pictures and, doctor, what is this mushroom and what is the importance and everything. So yes, definitely when you have mushrooms collections and when you take a photo, sometimes yes, I'm able to give you the genera but not the species level, because the species level identification needs right now with all molecular works. Then we can group them or narrow down them to the particular species identification. Okay, and then not only that, we do a lot of different types of protein coding gene and also ribosomal gene to identify what are the groups, and if it's really congruent, then we accept the phylogenetic species concept with the morphological species concept. So you have to know, because biological species concept sometimes we can culture, sometimes it may not have the culture. So it's very difficult to to work on the biological species concept. But with these two, morphological and phylogenetic species concept, we can reliably tell people that this is the right species that you can name it. So this is the process how we have been doing this for many years. And also now with the inclusion of chemical analysis, we can able to tell what are the compounds. For example, for mushroom poisoning, we need to know some of the highlighted compounds for medicinal, medical aspects, because sometimes people misidentify with the edible groups and then they confuse with the poisonous, and it causes a lot of problems. And the JKNS, Jabatan Negeri Kesihatan Negeri Sabah, they approached us for the identification. So we do a proper way of identification so that we get to know what are the main components that available in that particular mushroom that have been categorized as poisoning mushroom. So, with all the introduction, yes, I'm going to talk about the success story that we have developed for the past 6 years. Okay, so definitely for the past 6 years mean it's a lot of mushroom collections. I do not want to immediately say that this is a new species, this is a new record, without every region's samples represented. So we work with Singapore Botany Garden with Serena, where she always send me some of the samples, and it's a nice story to tell the whole Malaysia and the Peninsula, Singapore as well, and also the Peninsula side and the Borneo side, so that it covers the whole region, okay, including some from Indonesia. So when you look into this, okay, 1960s until 2000, okay, only 3 species was known to Malaysia, okay, the Tricoloma, the Pucena salsipes, and the Pucena speciosa. The speciosa was described in 2006, which was previously we know that only 2 species was available. But how many species are actually in Malaysia? We have no clue. So even though it's a common fungus, and every time when you are in the forest, you will see these guys very, abundantly. Wherever you go, you will find these species. Either they are hairy or they are non-hairy. Either they are pink or they are yellow or they are orange or red. So the colors definitely makes a lot of difference, because when you see a different color, you assume that it's a different species. Okay, but in this case, as you can see from here, they are lighter pink, and this is a darker pink, and this is a lighter orange, and this is a dark red orange, but exactly they are same species. Okay, and this one is a bit brownish, so it's a totally a different species. But then my interest was on towards this group, and me and my students and also my collaborators, we did collect all the specimens from each of the states, and also from Sabah and Sarawak, and also Peninsula and Singapore. And we categorize as that, as you can see from here, within the Tricoloma, even though we have tricoloma, we have one new record, the sinensis group. So the sinensis group is actually also was described in China actually. So the Chinese specimens was similarly correspond to the Chinese specimens, the Bornean specimens. So it's an evident base where even the morphology was very unique. The hair is a bit sharper and also longer compared to the tricoloma, okay, which is this group, but they are sister group to the tricoloma. So it is a neo record here. However, here in this claim, the Thailand region, they have currently they have labeled this as a new species. However, I don't agree with the new species name because this is actually a sulcipase clay, which is only, without a proper sampling, they label just because of the color. Okay, so we have to argue this in our current paper, which is coming soon, where for the whole region of Malaysia and Singapore and also Indonesia as well. So it will be an interesting paper for all of us to refer to how to identify the sinensis or the tricoloma, the sulcipase, or even the speciosa. Okay, so Borneo has all of these species, the 3 species, and additionally we found one new record, which is the Spukena sinensis. So this is what exciting for us. You know why? Because addition of species list for our country is the most important thing. How we do not know all these, because sometimes people see these hairy things, immediately their conclusion is tricoloma. But you see, recent in 2016, Philippines has recorded a new species of a similar way of hairy cup, which is referred as corfi. So this is interesting to go on with a lot of new samples. So as you can see here, the weird looking phenotypic characters of cup fungi is actually amazing, because some are hairy, very long. So I assume this is sinensis. I assume this is Spukena tricoloma, but we never know, because they are almost resemblance to tricoloma or sinensis. However, the yellow color groups, it shows like Spukena indica, but when we did the DNA, it almost clustered together with the Spukena sulcipase. So there is no differentiation. However, here this is speciosa with the dark, a little bit brown. It's totally. But this weird looking cup Spukena, we have identified this in the field, and we have currently generated sequences, and we will, verify this into any of these species that I listed just now in the phylogenetic tree, and you will be able to know whether they are just because of phenotypic difference or is it a new species. We never know. Okay, based on phylogeny, species concept, then we will verify that morphology and then the species concept for phylogeny. So that's all about Spukena. And then another success story that left out in Borneo, especially in Sabah, no documentation on these species apart from Peninsula Malaysia. Okay, Peninsula Malaysia has been domesticated this mushroom and has been very successfully commercialized this mushroom into a big large portion of decoctions and a lot of it. And this is also known as Khazana Negara, where after 1990s, I think Datuk Mahathir has cured his his coughing because of this mushroom. So it was a very famous news, in the news article. So everywhere was, and this is highly prized for their sclerotia. Okay, these are the resting structure of a mycelium, where they deposit here through the Eiffel, the Eiffel coil and everything, and they become a resting structure, either above ground or above ground. Okay, below ground. Sorry. Okay. So what is so important about these mushrooms? Definitely, this has been proven scientifically. They have a lot of medicinal values, including the asthmatic, especially the lung and the respiratory syndromes. All these, the decoction, the sclerotium, has been made it into powder with a lot of, chemicals, and then these are treated as anticoagulant, antiviral, and also to enhance the immune system. So this has been proven in many studies done by Mardi, there is a big group working on this on a medical aspect. So they have been, well established doing this in terms of, the benefits of this particular mushroom. However, the sample location and the sampling designs or map distribution has not been studied well. So what we did all the literature search on this species, the genera throughout the world, and then every continent, especially in African continents, we have many of the species over here, but they are totally different from the Asian species. However, in Peninsular Malaysia, you have 3 different species that was described by Dr. Tan. Okay. However, the Borneo region one, they were misidentified by Pegler as Amaroderma and then to Lichnosus, but it was not as Lichnosus. It was an Amaroderma. So that was a mistaken in one of the record that has been checklisted in Malaysian fungi. Yeah. Okay. Then Sarawak Yamashita, in his own sampling plot in Lambert Hills, they recorded that Lichnosus rhinoceros. So Sarawak has one record. However, Sabah do not have record all this while until we did this study, and we did extend the sampling for the past 5 years, and we completed how many different types of Lichnosus that are available in Malaysia. So when we did the DNA analysis, very exciting results where we did have cameronensis, we did have rhinoceros, we did have Phylogenetic. But these 3 species have been commonly known in Malaysia with this peninsula from Cameron Island. However, this one, as you can see from the purple, these are the Inensis, which is from China. It was described in 2016 or 14, I guess, from the paper of Yu Chengdai, where they found this new species and they described as new species. And we found this literally in a waterfall area, which is amazing to see that tiny structures with a very big sclerotia. Okay, so something exciting. Okay, so we have China sample here, which is just, it's not that surprising because tropical. Okay, the region where this was found was Hainan-ensis, is China. It's a tropical area. Okay, and then within the Lichnosus, we found another interesting finding where we found that all the Lichnosus rhinoceros that has been named synonyms, rhinoceros or rhinoceratis, everything is a huge complex group here. As you can see from here, why I say complex, because there is no definite character or there is no definite identification till the species level, because as you can see from here, some of the clades are forming a different clades here, are probably a different species, but it's not fully supported just within one gene. So what we are doing now is we are looking into the protein coding gene to resolve these clades. Maybe the Malayo Basin one is typically a unique clade, because when we check the DNA sequences, they are pretty unique towards just this location, the Danum and also the Malayo. So it's something going on there with the base pairs. Okay. However, we will verify this after we do the TF1, which is the protein coding group. However, we have Lichnosus, Tigris, and Hainan-ensis, Cameron-ensis in Malaysia, and definitely in this clade from the complex, as a similar study that we finished for Pukena, we reveal many species, many cryptic species. So it's not surprising if we find any cryptic species within this complex. Okay, so as a result for this group, we so far have 2 new records for Sabah, 1 new record for Malaysia. And these are the summary of this, where the Cameronensis for the first time we found in Sabah, which is from Cameron Island to Sabah. It's a 2 different region. Okay, and then first record of analysis in Malaysia. So as you can see, this is the only species that resembles the Chinese materials, okay, from China. And the complex groups, as you can see from here, the complex group possesses a lot of phenotypic characters, that variations, too much of variation. But as you can see, some are golden yellow color, and some are dark brown, some are bigger or smaller sclerotia. So these are the things that we want to look into it further, okay, to see whether they are one species or many species. Then the part 3 I'm going to talk about. Okay, so far we have we have talked about Ucena, the cup fungi. Within the cup fungi, of course, there's one edible group, which is the sulcipes. And the second one is a medicinal type of mushroom, which is one of the examples that we have made. And now, the story of poisonous mushroom. This is a very huge problem in Sabah, where we do a lot of collection base, and we identify them into different genera, because species wise we only have to wait until the DNA comes out. Okay, then we have to do the phylogeny, and then the chemical, then we combine all this information. Then it takes time, it's a long time, but we're able to crack all these mysteries one by one as per the cases that we receive. Let's say if it's a chlorophyllum molybdites, immediately we know what species is that, because it's now people are already aware of that particular species, because in Peninsula these are the things, these are the chlorophyllum cases, a lot of cases appear. However, in Borneo they are still looking for different types of mushrooms. It's not just chlorophyllum, but there are different types of mushrooms still people eating them, which resemblance to Termitomyces or termite mound mushrooms, all can be eatable. No, that's wrong concept. Okay, and also here they believe that any insects on top of the mushrooms, it's eatable. No, that's a wrong concept as well. So we have to make sure nobody, because so far we have at least 4 deaths, all these one, okay, from 2001 till 2019. So, but the cases are sporadic, but still a lot of variations between the mushrooms that we receive every year. Okay? And Peninsula side, Dr. Shin, she's looking after all the Peninsula side, and the Borneo side, me and Dr. Asnol from Sarawak. So we are collaborating between the mycologists and the chemists and also the medical officers. We are training them into all the, how to identify, how to collect them, fresh collection. Because if you're giving me the puke, I cannot identify the puke, because it's the mushroom is already digested or destroyed after the eating, and then we can't able to identify them. So that's why I say all the medical officers, the the inspectors, medical health inspectors, they usually go to the site and then they bring the mushrooms for us. So when you look into the overview of the poisonous mushroom incidents for the past 1990 and 2019, okay, a lot of countries have recorded any different groups of mushrooms, okay, but sometimes some are lethal, some are, like for example in temperate forests, usually people encounter the Amanita species. But what about Borneo? We have no data. We have no data that tells how many sequences, how many DNA sequences were available. Nothing. So we initiated this project in 2015. So slowly it's evolving, and we have some cases where we have stopped, in a one particular district or different particular district, because there are many districts available in Sabah where each district they have different types of mushroom every year during the rainy season. So to avoid that, we need to do the awareness and things. As you can see from here, this is a this is the Podocarpe, or the Podostroma we call. This is actually, in my own experience, you cannot even touch this, because you know why? This is highly poisonous. Okay, but it's available in Sabah and also in Peninsula and also in Singapore. Okay, so these mushrooms, whenever you see, please do not touch, because even when I do the cultivation, in the culture, you can feel the dizziness, okay, because of the spores. Because this red color It's all like a powdered form. So whenever you touch there, the powder will stick into your hands. Okay, so you be careful with this type of mushroom. Okay, these are very lethal in Japan, and a lot of death cases in Japan due to this mushroom. Okay, when you compare with temperate mushrooms, these are the type of mushrooms, and still you see this Podostroma, poison fire coral they call it, and they have this a very little point of amino acid kind of structure here, triclosine. Okay, this is what causes the human being to be, when they eat mistakenly, or when they touch, or they inhale, or they expose. This is what going on. Okay, so there are many other types of mushroom, but these are the names have been categorized. Chemicals have been categorized. But what about Malaysia? So this is what we have to address, and everybody need to be like, work in a group of citizen science. When you spot a mushrooms, yes, take a picture, and then you can ask for help for the identification, or bring to the lab where the University of Malaysia Sabah is always open for you all. And this is how we should work together. Okay, like sometimes we have to help each other to eradicate these mushroom poisoning cases. So these poisonous cases, definitely we are lacking a lot of sequence from here, and also a lot of compounds that we haven't characterized for Malaysia. And recently, what we found that in 2019, when I was in Kota Marudu, the neurotoxic mushrooms groups are people eating these because they grow on termite mound. Some of them, as you can see from here, the number 4, which are like the pinkish, and it's ambonate kind of caps, and they are very beautiful, but they're growing on a termite mound. Just because they are growing on termite mound, people assume that it's edible, like cendawan busuk or cendawan taun, like the Termitomyces, but it's not. Okay, so make sure you are not eating the wrong one. And this is the first occurrence for Sabah, which 18 cases were spotted. All the 18 cases out of the 2 were admitted in ICU, and treatment were done, and luckily those 2 became normal. Okay, so, and when I analyze this mushroom, it seems like very closest to the Mastoidium from China, which is Borneo one. So these are the shapes of the termite mound mushroom over here. As you can see, this is a termite mound. We found a lot of species, and this species, Antholoma, is something very, very dangerous. Very dangerous. So do not consume this. And when we analyze the chemical, these are the chemicals. As you can see from here, hydrotoxic tryptophan, which causes insomnia, headaches, muscle spasm in mouth. So this explains a lot, because one of the patients have described muscle spasm in mouth. So it is relevant. Okay, and also they attack the cerebral enzyme system, so maintaining the normal function of the nervous system. So that means this is one of the enzymes that are helpful in neurotransmission. So the 2 patients were in ICU, unconscious. So maybe due to the specific type of chemical compound. And some can cause seizures. So these are based on LCMS analysis. So it's pretty much good info for all of us to educate ourselves not to eat any without any confirmation. Okay, so you have all the mycologists around Malaysia or Singapore. So you have to ask first. Even the hikers, even don't assume that that it's an edible mushroom. Okay, even if it's an edible mushroom, we do not know what is on top in the environment. Sometimes you may end up with leptospirosis because a lot of mouse that urinate on top, or sometimes even any other kind of bacteria growing on top. So we have no clue. So this initiated us to work with University of Malaya and University of Malaysia Sabah, UNIMAS. So we come up with a lot of activities, general activities like mushroom poisoning, to make people understand how to identify the, not to say the prominent characters, but at least the general characters to differentiate whether it's a poison or whether it's a non-poisonous. Okay, at least people need to know which are the groups that is. Okay, so these are the kind of reports that we do for the medical people at final cases to wrap up all the cases. So this is a kind of simple identification and also molecular and also based on morphology. And then definitely environmental awareness for public, including school programs, teachers training, staff training, and also outsiders to learn all of the differences between these mushrooms. And the interesting story now, which is the success story of a glowing mushroom. Okay, glowing mushroom is a very interesting mushroom, which is also under the macrofungi, where, as you can see from here, these are leaf litters. Especially in your eyes, this is just leaves. Or in my eyes, I can, imagine how the mycelium covers the whole leaves litters, how they degrade these leaves, and then you can see how these mycelium grows. Whenever this leaf litters grows, it involves the mycelium. Okay, so this is a fungus that my friend Serena spotted in Singapore as well, Neonothera panus nambi. So molecularly, we have identified these. They are quite beautiful as well. And then, in total, we have now about 32 species of glowing mushroom, which are some of them we have already successfully cultivated. And this is something unique, okay, something unique to see, because they can glow either the whole body, only the side, like this one, or they can glow only the mycelium or even the wood. Okay, as you can see, this is the glowing version of this one, Phylogenetic. And my interest is to utilize these glowing mushrooms into how to grow them, how to develop them, how to the glowing intensity, how to increase them. So we need to come up with a lot of cooking recipe. So this is how we do it in the lab to increase the intensity, as you can see from this A till G. Okay, so these are the intensity of these species for the glowing, which different media formulation. We manipulate the formulation and we can create how intense is the mycelium and to glow them. Okay, so as you can see, sometimes the leaf litters glows very beautiful. Okay, this is the sample from Singapore. Okay, and apart from that, I would like to introduce a lot of different types of wild edible mushrooms. So this one, you might have seen these in field, okay, whenever you go for hiking or anywhere. These are all the edible treasures that we have. But what are we doing then? Okay, are we doing any research on these? Are we contributing for at least any kind of taxonomy work? No. So that's why it's a lot of work to be done. If you are working on specific group, then it's easier. But if you're working on a general base, it takes a longer time. So that's why I finish one by one each group. At least takes 2 years. Okay. So with a lot of samples, we have a lot of representatives, and also we can do a lot of microscopic. We can do a lot of DNA studies to identify them, and then if some can be cultivated, then we cultivate. That's all. We keep the germplasm. That's it. Okay. And keeping the germplasm is one way of conservation. You don't go and destroy them again and again and again in the same area. And this is one of the PhD topic that I've done. This is we call the Unkugan, Lentinoid, and Mangkugan. All these different vernacular names. They look so different. Phenotypically, they are different. Okay, but it turns out to be only a single species, which is the Lentinoid. Okay, and here, as you can see, so many variations in the phenotypes, but it turns out to be the same genetic. Okay, what we can tell about this, because people have categorized this group at least 136 names, synonyms under this group. So you see how morphology can also leads to confusion. So now with all the molecular, we are now very sure these are all similar species, not at least different species. Okay? And this is what I've talked about just now when the beginning, how Corner has segregated these based on 3 different genera. Because Lentinus, Panus, and Pleurotus, these 3 genus has been always problematic genus. Okay, because they look like oyster, they look like very Pleurotoid kind of thing, or Lentinoid. So like you see from here, these are from Amazon collection. So you see how inward or outward inroll margin here. Okay, and then you have also some of the different kind of base over here, and then this is another type of, because Peninsula contains this a lot. Okay, Peninsula, Malaysia. So this is a work that I have been continuing for the past 6 years, and we are almost completing this next year. So with a lot of samples from different continents, including Russia, because we have collaborators send us a sample, and we are analyzing the DNA for all the continents. So this will become a very major paper for us to indicate all the regions are different types of species and how segregate they are to tropical and temperate regions. And the golden cantarelles, of course, it's not just in Northern America, tropical Africa, or tropical Asia. Okay, we found this actually in Sabah. As you can see, there are many different types of cantarelles available, and in Peninsula there's only one paper indicating about 6 species from the Paso area, but different area or different forest type have not been explored. So it's the time for you all to spot down all these kind of beautiful mushroom, and you can send it to us, all the herbarium. So from the forest to the table, how we can culture, so that I don't have to go back again to look for the same organism. Okay, this way I conserve those species. Okay, and also these germplasms need to be protected, because, for example, you have rice cultivars, and you have so many rice in a different country, and India is one of the best example where they have all the seed banks kind of things, germplasm of rice in different according to the different districts. So it's the same way of taking how mushrooms also need to do. So whatever the culturable species, which is a saprophytic fungi, it's easy. But ectomycorrhizal, it's a bit harder, but you need to have a regular or routine way of cultivating them. Okay, because sometimes ectomycorrhizal nowadays is becoming easy to cultivate. For example, Cantarellus in Japan has been cultivated. Truffle, morels has been cultivated in different temperate area. But it's the technique how you deal with that. Okay, so definitely one day we will have a good collections of all of these mushrooms for the next generation to continue their studies. So here there are some understudied groups, which are my favorite groups, which are the wood decaying fungi and their beauty. Okay, they are the pretty jewels of the hidden jewels of Borneo, because each of them possess a different hymenophore, which looks like a beehive. Okay, and somehow you can see how beautiful their pores are, and they are hairy. Sometimes they have so many hymenophore types. Sometimes it's like look like the beehive. As you can see, there are different types. So these types, what am I going to do? Definitely, I'm going to do on the culture forms for enzymatic studies, because these groups are enzyme properties are higher for these and caseous enzymes like lignase, cellulase, zelanase, hydrolase enzymes. So there are many different biological, biotechnological application can be used. So we just keep collecting for Sabah and keep preserving these genetic resources for future uses. And the last group that I would like to introduce is the zombie fungi, which are, of course, many people have been, many people been understanding this capable of causing disease, which is on the ants or any other insect groups. So these are the collections of from Borneo. Still awaits of a lot of identification and characterization, and these are sometimes the trade-off of these zombie fungi. We need to understand why does this happen to only in ants group, or is it common type of other host. So like for example here you can see on the spiders, okay, these are spiders, and then sometimes on wasp, sometimes on cicada, and mostly on ants is a similar way of interaction. So it triggers my interest to look into it further with culture base. So we are working towards on it, and if you all see one, yes, definitely you can contribute to IPBC's Borneoensis collections. And sometimes we have to, educate our students and people in a simple way. So we create this kind of posters with my student. So these are the things that we do in our lab, not just focusing on the DNA, but it involves like the whole, the whole community level. So the public outreach and the conservation awareness, this has been stopped for 2 years because of the COVID, but I hope to develop this again after this, for this year or next year. We can have this for, for Sabah, or even in your region that you are coming from. You all can actually simple kind of, public outreach you can do for mushrooms. Okay, but you must get a permission from the state park and everything, and then you can have such a wonderful activity, because each of these activities will contribute to the scientifically and also environmentally. So, and also for yourself. Okay. And sometimes we also document all the traditional knowledge and the traditional based food that prepared using the mushrooms, local mushrooms. So these are the way of how we integrate our studies on mushrooms in Borneo. Okay, training, a lot of training for rangers. So if you find all the rangers in Sabah, definitely they are trained. Some of them are really good because each eye works differently and each mind works differently. So when it comes together, it's amazing to see the data. So that's what interests me, to work with locals and also internationally or even nationally. So student activities, we can do a lot of different kind of mushrooms character, but you cannot teach them the complex way. It has to be simplest form. So we have our module. We have prepared our own modules to teach them, to educate them, and we can share these with our own students and local students or international. So these are the activities, not only just throughout the forays, but even in the classroom, how they can make interactive class activities using mushrooms, or when we are studying about biology. So we are still looking for mushrooms. So it's always a great journey to explore more into different places and different people that we are meeting, and it's always the guides that I would like to thank them in Tab Sabah, yes, and Sabah, and even the entire team on the forestry and everyone, because without them I cannot access these places, even though hard, but we managed to get a lot of samples. And I would like to thank all my collaborators here in this talk, where these are the collaborators that we worked for the past 6 years, and we initiate all this give and take policy, and then you have to win-win situations sometimes. So these are the kind of network that we created for mushrooms in Borneo especially. And these are the local students and international students that have been the green one, all graduated. Some of are ongoing, some of the incoming. So it's a growing kind of effort that everybody is working towards, okay, to betterment on the mushroom farm. So with my motto, I always have this: given the opportunity, we create a community. So thank you all. So if you have any questions, please, I welcome all your creative and constructive comments. Okay, thank you.
Thank you, Dr. Jaya. It's very amazing to see so many types of mushrooms. I have never known there are so many, and it's a very interesting findings. And I really like the glowing mushroom. I always hope to see one at least, but among all my jungle trekking activities, no, not my luck. But anyways, before we go to the Q&A session, there's a housekeeping announcement that I want to repeat, which is the donation. For those who are on Zoom right now, you can donate to Sabah Society by scanning the code just at our background, Dr. Jaya and my background. On the left side here, there's a code that you can use the Do It Now QR scanner or Boost to donate to Sabah Society. And of course on Facebook, you will see this. Hold on. You will see this poster that is donate here. You can scan the same QR code using Do It Now or using the Boost app to donate to Sabah Society. Now, Q&A. Are you ready?
Oh, yeah.
So the first question here is how to become a citizen collector.
A great question. Citizen collector. So it depends on which region you are. Okay. Communicate with the mycologist in that region. Okay. Then you have to establish a proper way of collecting. Like for example, let's say if I'm getting a sample, definitely the mycologist will teach you the guideline, like how you can take a picture. Okay. So you can consult with the mycologist where they're able to tell you the proper way of collecting the specimen without any harm or without any contamination. So that's more important for mycologists when they receive the sample for identification purposes. But always for citizen science, usually you all can take good photos, a good description, like when you see what color, when you see the bottom part, what is that? Okay, all these kind of things, it makes you learn more, because I've seen this literally in the United States where even a common man, they will be telling that every details in their own language, which is not scientific, but still it's useful for the mycologist.
Now here Rachel also continued to ask, like, how do I reach out to the local mycologist?
Oh yeah, that's a very good point. I mean, here, as you can see from here, from this diagram, I mean, you all can contact or search in there, but these two professors already retired, so we are the younger mycologists, so you all can approach us. So I'm still young, I guess. Okay. So we can actually communicate which are the regions. So like for example, in Peninsula you have many. So in Peninsula you have a few number of them. So Dr. Shin is actually from Peninsula, and even Singapore, Serena is one of my good friend. She is also within the Singapore region. She is the one person that you all can communicate. And also Associate Professor Shin within and Dr. Ross is from Penang side and Kedah, Perak, all these, and actively working on towards the collection base is Dr. Sumaya as well. You can communicate, or Dr. Muhammad Asnol from Sarawak, and me in Sabah.
Thank you. And the next question is how to correctly collect a mushroom sample.
How to correctly collect. Okay. So, correctly collecting is something that word is not appropriate. But the thing is, you can collect as you want. Just a simple bag, paper bag, or plastic bag. Also, you just have to, if it's a root kind of thing below ground, then you have to shovel it until the root level. Like for example, Termitomyces. They have a very long pseudorhiza, which is like a long stipe. So you have to dig until you see that. Then I can tell you, oh, this is a Cendawan termite. So those are the things, tiny characters that you have to. And then for general fungus, usually on the wood, it's pretty easy, where you just collect them from the surface of the. But always cut a little bit of a wood so that I know it's from the wood collections. Okay, if it's soil, you make sure that you dig out a little bit soil and then you put it in the, in the collection bag. Yeah, it's something that, but I wouldn't suggest this to all the hikers. Like you can go and take out all the things. No, because in Sabah, in Sabah we have a pretty strict rule. But if it's in your backyard or sometimes, we never know, because while I was driving in UMS, I found the new species. So we never know. In your backyard or in your garden, you may find new species, which we do not. Yeah. So any type of fungus, if you find it interesting, or take a photo, send to me, and I can actually able to identify, like to genus level. Then if it's really important, I will tell you, okay, please come back to my lab. Come to my lab with a sample. Then definitely, I mean, if it's a very interesting group, definitely I will let them know.
Yeah, I missed the chance already. I came back from my trip. I always snap picture of mushroom because I personally like seeing mushroom, because there's a lot of types, white or black one, but of course I don't know any species about it. Yeah. So the next question. Why are Amanita section Caesarea, shown with the poisonous Amanita species? This is from Curtis.
Okay, thank you, Curtis. That question is a very specific question, but I would like to answer as overall for the Amanita. Amanita group is actually all of the species. I'm not surely can say that it's edible, because until now it's really, I have phobia with Amanita because a lot of cases on Amanita. Yeah, but in Malaysia you have this Amanita phylloides. It's the most number of cases that we have spotted. But the Caesarea shown in the poisonous Amanita is actually not just the Caesarea, but the rest of all the Amanita. Because you know why? Amanita has the compound called amanitin, or even for the tropical species, we have no clue what are the type of compounds, the poisonous compounds available. Because every time when you talk about Amanita, is the muscaria, which is only occur in temperate region, which are the red color smurf kind of things. Okay, but in Malaysia it's a white thing and the yellow thing, except for the Amanita malayensis, which are the white to yellowish. You can eat according to the sources, but until now I don't encourage people to eat any Amanita species. Yeah, because in our culture, the traditional knowledge of mushroom is very lacking. It's not like Poland. Poland has a long tradition of mushroom knowledge, and also overseas, other Europe, North America, even in Southeast Asia, it's very less. But China, yes, definitely long tradition of traditional knowledge, and also Japan, but not in Malaysia. Malaysia, our older people, I mean the elder people, they have identified like Kodok, there was the Pulat Sisir, the general ones like termite mushroom. Even the termite mushrooms now becoming a problem, because sometimes they are mistakenly eating other types of, that growing on the termite mound. Yeah, so it's a bit difficult for us to tell them which are the edible, which are the poison, because there is no definite character. So like in Amanita, I would say, for me, all the Amanita, it's not great to eatable, but according to the Caesarea, actually not just the Caesarea, it's most of the groups within that are poisonous. Yeah, I hope I understand. I mean, I have answered correctly.
Curtis, yeah. So I hope you get your answer, Curtis. And the next question is there any rule of thumb way to identify edible mushrooms aside from proper indications?
Rule of thumb. That's why I say. Luckily, I mentioned this earlier, right? Because there is no definitive character to indicate whether they are edible or whether they are poisonous. But now with poisonous mushrooms, there is a chemical, but I'm not sure which chemical. It's still in the process of making it, where when you put that mushrooms in the chemical, it turns colorimetric. Then they're indicating the presence of amanitin or any other groups of chemical that poisonous. Okay, but for edible groups, it's based on your experience, based on your passed down knowledge from the traditional people, like our grandparents.
Yeah,
that's the only way, because we do not have a definite character to indicate that is edible group. Unless you're very sure, then you can eat it live. Then try it. Let's see tomorrow.
If not, send to your lab only lah.
Yeah, exactly.
So perhaps it would be helpful to produce a book on edible mushroom if they are poisonous, or toxic.
Actually, the group and my group are developing the wild mushrooms. So it takes long years, to get all these pictures and all the description and all the DNA stuffs. So it's not easy, but the thing is, it's coming in different volumes. Like the group, they are preparing all the marketable products of commercial mushrooms, which are all the edible ones.
Yeah.
So they are focusing on that, and my groups are focusing on the wild mushrooms of Southeast Asia. So it will be helpful in the coming time for hikers to see and also feel that how the colorful mushrooms or the glowing, because the glowing mushroom is kind of interesting to feel, to see them in the night. But at the same time, at night, all the nocturnal animals are there, especially snakes. So I have encountered many times, but luckily we are going in a group, so it doesn't happen any accidents. But sometimes when you're going, in a group, then it's okay lah. But if you're going alone, that's risky lah. Don't take a risk, okay?
So what are your most challenging, trip or findings, or what are your most challenging moment?
When you encounter cobra, that's it. Yeah, when you encounter cobra, you just become silent and back out.
Oh no. So there's a question from Amy Chung. Has the black or white truffle been successfully cultured?
Yeah, you see, that's a very tricky question. But truffles, truffles are actually now being cultured in. I read that Europe. There's one professor has successfully cultured this truffle group, but it's not like well established yet, like morels. So I think it has been cultured, but the thing is, it's not been that famous yet. So I hope maybe we can track down that information somewhere in the internet or Google to find out that the truffle group, because I remember one of our collaborators from Germany has indicated that the truffle has been successfully cultivated.
Okay, hope you get your answer, Amy. And is there any plan to develop a mushroom growing kit for glowing mushroom or edible mushroom?
Okay, edible mushroom, definitely. Any types of medium can be formulated to grow them. For glowing mushroom, it's a bit tricky because they need the intensity of the glowing different in terms of the substrate. So when you create the substrate, it's not just a common substrate. It has to be very promoting that glowing properties on that substrate. So we are formulating that type of things for our work, and let's see. Hopefully, we will be successful in creating a nice glowing media. Yeah.
And Curtis has another question. Do the pillars and type of Ignosus. Oh my god. Sorry.
Yeah, Ignosus.
Species. Yeah.
Okay.
Correct, right? I'm so sorry if
Very
I butchered the name. The species name. Yeah, species have any medicinal value as well.
Yeah, actually, Pileus and the types, usually people have screened for antimicrobial, which are like antifungal, antibacterial. But the major part is the sclerotia, people looking into the medicinal value. But not, as Curtis says, the Pileus and type. But there are some papers, Pileus and type has shown very minor activities of other activities, not just the coughing or asthmatic, because the sclerotia has the most number of activities. The active compounds are mostly found in the sclerotia. I hope I answered.
Yes.
Yeah. Any questions?
Yeah. For now, let me check on Facebook if there are any more questions.
One question here.
Yeah, you may.
All right. Interesting fun stuff. Can you hear me?
Yeah, how are you, Chun?
I'm Chun from Tower. Oh yeah, we have met that day.
Yeah, I know you.
Yeah, first time attending a talk. So I'm just wondering. So for example, some wildlife species, they are going extinct because of hunting and poaching, including plants. But I'm wondering, are there fungi species that are very few and very hyper-endemic that collecting can threaten the population?
Yeah.
And maybe a second related question: have there been any cases of fungi species that have gone extinct because of human activities?
Yeah, that's true. Actually, thank you for bringing up this matter. Actually, yeah, as an iconic species like elephants, wildlife, everything is extinct and stuff. Definitely, in certain mushroom groups, we do have some extinct vulnerable. We can say vulnerable because the kind of gastro type of mushroom, the gastroid forms, are very limited survival because they are reproducing. The reproduction is very limited because of their closed. They are not like the regular mushroom that has spore freely can, wind pollinated or animal pollinated. But these gastroid things are very well protected, under the ground. So it's very difficult to find also. Even when we do the sampling, those are the things that, we find very rare. So some of the species have been listed in IUCN, and there's some species that in Sabah or even in Malaysia, Lignosus is endangered because too many harvesting. Okay, they proposed as an endangered species. And there's another species called Baglososporus, which is going extinct. But without proper monitoring all the field works, you cannot say that it's extincting. So from our surveys, I would say it's limited. Yeah, because in Sabah, still we are really good with forests and stuff. So the species are still present so far, those proposed in the IUCN, and because they cannot propose like animal and plants extinction or vulnerable or threatened, because for fungus it takes a long time to prove whether they are really, like for example in Tibetan cordyceps, it is extinct because that one is based on the Chinese, all these Tibetan area, they over harvesting occurring, so it's declining. Definitely there's a trend, and that's been as proved in Proceeding National Society of America, where the PNAS, where they have indicated in their paper that Cordyceps sinensis or Cordyceps militaris is actually depleting in the population. So mushrooms can be also going extinction, but we do not know. But there are many other types of mushrooms that we can see, but the things, how frequent you can see them in the field in a different areas. Like for example, I have one group of mushroom which it was described in Danum Valley in 1994. I cannot find them after 2002. So that means the time interval 2012 actually I found again. After that, I cannot find that until this year.
Mm.
You see, that means it is occurring because it is a Dipterocarp associated Pleurogaster we call. So, as from my experience of field work, okay, we find this very big interval, 10 years, 10 years. So, what is going on? Why I cannot find that particular mushroom in the same place or even a different place? I cannot find them. And some more the host is Dipterocarp trees and stuff, but I cannot find them. So, what's going on there? So, that's why when we do the recording, all the documentation, we have to make sure which are the species that can be occur only at that particular time and the month and also the season, everything. Because based on the trend that we have, then only you can see, okay, very clearly this population is declining. So like the green Pleurogaster, something that I would say very rare, because it took 10 years and then 10 years. So it's a very, very long year. Sometimes my other mycologists agree with that theory where some mushrooms need to be looked into the very detailed, but mushrooms are very difficult and unpredictable population under the ground, and then they pop up and then they go. That's it, they decay. So it's very difficult, but we are monitoring every year now. So let's see which are the type. But definitely Pleurogaster is one of the mushroom that it's already listed as vulnerable. Okay? So I hope I have answered all the IUCN matters on the fungus, but you can also log into the fungal IUCN, the red list, and you can able to track all the types of species according to the country. So you can look for Malaysia, then you have all the list of species.
Okay. Probably I will get the website from you again, and then so that we can post it on Facebook.
Oh yeah, the fungal red list actually, fungal IUCN red list. They are all doing the proposal for Malaysia. Not only Malaysia, for the Southeast Asia.
Okay. And we have some questions, new questions from Facebook. What kind of mushroom most related to your research in Borneo?
What kind? Can I get the
what kind of mushroom are the most related to your research in Borneo?
Oh, in Borneo. Okay, that's a very good question. The most connective way of I'm passionate about is all the Lentinoid and Pleurotoid, which are my babies, because those are the things that I would say my babies, because I started with the molecular and taxonomy with my supervisor beginning from that group. So I'm still have a lot of interest in that group. At the same time, I want to explore those very major importance one. So that's why we are expanding the research into other groups like gastroid forms, zombie fungi, and also because these are all lack of data available for Malaysia. So we have to address those problems first. Because when we always ask how many plants, we are able to tell 2,000, 30,000 plants, and how many animals. Okay, these are the primates, these are the birds. But when we asking a question, how many fungus, how many diverse is mushrooms population in Sabah? That's the question mark. Even Malaysia, it's a collaborative work that we all have to do. That to document these are the kind of work, as an overall group to indicate. Oh, Malaysia has at least 4,800 species, something like that. Based on records, definitely it's something that it's like not even a 20%. So it's a long gap, for us to address all.
So this question earlier is from Sam on Facebook, and we have one more question that all right, from Malaysia Sue. What about magic mushroom? Is it available in Sabah?
This is a very interesting question, right? You see, everybody will ask the same question in every talk. how about the magic mushrooms? I want to know. Okay.
Yeah, that's why I started.
Yeah, it's actually interesting. It is really interesting because now it's becoming a medical aspects of uses, like instead of using for other entertainment, hallucination and everything. But still, we can't reveal those information. But definitely, you all know that people have misused these resources. Yeah, people have misused these resources. Even there was a lot of articles coming out in the Petri Dish paper magazine where in youngsters in KL they're using this and cultivating this from they're buying it from Amazon or even from Shopee.
Oh.
Because the what do you call the spawns, you can get it from online. So it's a wrong thing to do because it is categorized under narcotic, if I'm not mistaken, because you cannot carry those hallucinogenic mushrooms collections anywhere from a different region. So it's illegal. But as a scientific way of categorizing the name and species name and everything, yes, we are doing it, but we are just not revealing it. That's all.
All right. I hope you get your answer, Leisha, soon. And let me check one more time. Is there any? Oh, I think there's one more on Zoom. Alien mushroom. Okay, from Chungsing, and Chungsing asks.
The name.
Is Chungsing also asks, is consuming wild collected magic mushroom it's legal?
Yeah, definitely it's not legal, I would say, because I mean it's not something legal here.
Yeah.
I mean,
all right.
Yeah, I mean if you try class it as a narcotic, yeah, exactly.
Yeah.
It is actually, but I was surprised too when I was reading.
Okay. Any more? I think that's the last. Is there any more questions from Zoom? Maybe I give another chance before.
One more.
One more question.
Yep.
So I'm just interested because of all your photograph compilation just now. I did not see any. Do you know the bird nest mushroom? Nidularaceae.
Yeah. Nidularaceae. Because those are very least group that interesting to us because it's a common thing. It's a common thing, and the species wise is not so interesting because the species is have been identified. Many people have been identified, and I don't see it so exciting for me. Did I answer your question, Chun?
Yes.
Yeah, but
just
fungi is unique.
The way of
it's a unique fungus, but people have already studied that so frequently, so I don't want to repeat that.
Okay, so thank you, Chungsing, for your last question for tonight. And thank you so much, Dr. Jaya. I learned a lot, and I hope everyone here on Zoom and Facebook also learn a lot. I see a lot of your friends are on Facebook, yeah. And of course, just now I saw that you do some events as well, educational events. So just letting you know that you can always send it to Sabah Society, and we can always help you to publish and promote as well. Yeah, cross promote is always good.
Yep.
We support each other. Yeah.
Definitely.
All right.
Yep.
So that's all for tonight. Yes.
Yep. Thank you for giving such a very wonderful romantic night with me.
Yeah, you make it interesting. Yeah, you make it so fun and interesting and so colorful as well. Yeah, thank you so much for all that have joined us tonight. Thank you for your time, and thank you, Dr. Jaya, for your effort, and of course, thank you, Rachel, for helping me behind the scene. And yes, have a good night, and we will see you guys in our next talk by Jared Abdul Rahman. He will be talking about an art. Hold on, I will find the full name of the talk.
Thank you, Rachel. Thank you all for attending.
All right. So Jared's talk, the next talk on Sabah, from Sabah Society is about artists and ambassadors, representation and misrepresentation of Sabahan identity in art. This is by Jared. I hope every one of you join. Dr. Jaya, you can also join.
Yes.
Yeah. All right. So see you guys in the next talk. Bye bye. Good night.
Hello. Yeah, I found one more. I have one more question here from Leisure Su. Those glow in the dark mushroom, is it edible?
Recording stopped.
Okay, I think that's all from us.